All right, I'll give around a minute left and we can get into the lesson. All right, everyone. So it's 2:05, so we can get back, or we can just start with the lesson. I will get onto the slideshow and screen share. Yeah. Oh. All right. Just to confirm, can someone type in chat if they can see my screen? All right, cool. That's good. All right, welcome to day 2. Oh yeah, and once again, if you have any questions, please put them in the Q&A unless I tell you to do so otherwise. So yeah. 'Cause it's easier to check the Q&A than chat. Okay, so we're on day 2 of the Kid Teach Kid biology lesson. So we'll go into genetics now. First let's do a recap of the last class. So in the last class, we introduced biological sciences by focusing on the smallest and functional units of biology, or the cell. We also talked about biochemistry and dabbled a bit of organic chemistry, just to show like how the chemical principles that affect stuff like chemistry you learn in other classes, even physics, can all come together to produce order and structure for biology. And we also talked about the macromolecules, which serve a litany of functions in our cell. In particular, in this unit, we're going to be focusing on genetics and how these traits are going to be passed on. At the next slide. Oh, never mind. And how the traits we talked about in the last class, for example, will be passed on to certain offspring. And we'll focus more specifically on a certain macromolecule, or a DNA, or the DNA, RNA, and proteins. And we'll talk about how proteins are actually made later on in the class, and also how DNA is replicated, as are really essential to genetics. So when we first want to talk about genetics, let's talk about the origins of it. We have Gregor Mendel, who is credited to be the father of genetics and founded a lot of the major principles that guide how geneticists actually practice in the field. So what happened? He took a keen interest on pea plants whenever he was doing his research, by which the different mating pairs that happened, or crosses, would produce varying outcomes in the progeny, or the next generation. For example, you would get 2 different pea plants, cross them together, or mate them by taking the pollen of the male plant, putting it to the pistil of the female parts, and producing a new seed which would grow to a new flower. In that sense, you'd also study their traits or characters. So characters are the heritable features that vary amongst the different individuals, and they analyze the resulting traits. A good way to think about the difference between characters and traits is character would be like height, and a trait would be tall or short. And the example I gave here, and which is what Mendel also studied, color could be a character. So therefore, purple would be a trait, and white would also be a varying trait of that color character. And because it's a character, we're also going to see that it can be passed on to future generations. The next bullet point here says that Gregor Mendel used true breeding pea plants. What do I mean by true breeding? So let's think about this way. If we have a purple plant and we cross it with itself, meaning we take the pollen from that plant and put it on the female structures of that plant, or the pistil, then it can produce another plant. And if we keep doing that, infinitely, it will always, or almost always, produce a purple plant, which means that it's true breeding in that sense, because it always will produce its purple plants when self-crossed. Similarly, a white plant that would be true breeding would always produce white plants whenever we self-cross them. So whenever Gregor Mendel wanted to study these plants, he always used a true breeding pea plant to eliminate any variation. If, let's say, there's a difference between the different traits that were expressed in a plant. So let's talk a bit more about Mendel's experiments with the pea plant. So he crossed a true breeding purple flower with a true breeding white flower. This would be called the P generation, or the parental generation, since the first one in the cross. The next generation, or the offspring, is called the F1 generation. And in this case, when Mendel did it, it only consisted of purple flowers. If Mendel stopped right then, he wouldn't have been able to find out a lot of the key genetic principles that he would have discovered if he continued, which is why when he crossed these F1 flowers again to each other, it resulted in a 3-to-1 ratio between purple and white flowers. So what did this mean? What happened was, at the time, the prevalent hypothesis for how genes, or not genes, but for how heritable characters were passed on to offspring was a blending inheritance hypothesis, where different traits would just blend together. If the blending hypothesis was true, then therefore Mendel would see pale purple flowers rather than just only purple or white flowers, as the purple and the white flowers would mix together. So Mendel believed that in the F1 generation, the white trait wasn't gone actually. So in the F1 generation, remember that it was only purple flowers, even though we crossed a purple flower with the white flower. He believed that the white trait wasn't gone forever, but rather it was hidden inside of the, but it was hidden or masked by the purple trait that it received from one of the plants, meaning that the purple trait was dominant to the white trait, and the white trait was recessive to the purple trait. This derived Mendel's model, which had a lot of parts to it, but the most important part of Mendel's model is a law of segregation. So before we talk about the law of segregation, Mendel also believed that there are two traits that each organism carried, or alleles, which are the variations of the different traits or genes. And he believed that one of them would be passed on. What did I miss? Nothing. We're just talking about Mendelian genetics right now. So if you miss anything, by the way, the slides are going to be posted on the website. So yeah, we're talking about. So what happened was Mendel thought that there was two different alleles, and one of them from each parent would be passed on to the next offspring. So in this case, since we have a true breeding, let's say we had like a true breeding flower. Let me see what the next slide is, if that has it. Okay, it'll have it on the next slide, but we're talking about Punnett squares. What happens is one of those alleles will segregate from the other one into a gamete. And these gametes, there's a male gamete and a female gamete, then they'll fuse together to form the new offspring. And then from there, we can determine what the traits will be. So that's the law of segregation, how in a certain like parental cell, when it's passed on to offspring of the sex cells, only one of those chromosomes is going to be, only one of those traits are going to be passed down. So how do we visualize this? Well, we use a Punnett square for this. So how do you find out which trait's more dominant? Okay, I think we're going to get to that soon. Yeah, we're going to get to that in the next slide. That's a good question. So you can figure out what trait's dominant by doing some crosses. So in this case, we have two plants on let's look at the top right one real quick. We have two plants, both are purple flowers. And when we cross them together, let's say we say the capital one, or from the previous crosses, we knew that since purple was masking the white flower, when we had a purple flower mixed to a white flower, we know the purple one's going to be dominant in that case. So I'll get to what dominance actually means, but for now, suffice it to say that the dominant one is always going to mask the recessive trait. So in here, when we make a Punnett square, we know we have two purple plants. In this case, we already knew some background information that they're heterozygous. Heterozygous, which is on the next slide, but it basically means that you have two different alleles or two different variations of the trait. In this case, we have the capital B and the lowercase b. The capital B represents the purple trait, and the lowercase b represents the white trait. So if we see here, we have these two capital B and lowercase b ones, and when we cross them together, you get one plant, or 1/4 of the plants, being both capital Bs, so there will be purple, since they only have the purple trait. But then we see something interesting. We see two plants that have a capital B and a lowercase b. But the thing is, both of them still have the purple trait. The reason goes back to what Mendel said, how the white trait will be masked by the purple trait. This means the purple trait is dominant to the white trait, so it will still be purple. But then if we have two lowercase alleles, the lowercase b and the lowercase b, and there's no purple and it's just the two non-functional ones, it's going to produce a white trait. So with these Punnett squares, you can pretty easily model how the different crosses are going to work out if you know the genotypes of the parents or the genetic makeup of them. And it can further on be used for multiple genes. As you see on the left picture, you can put 2 genes there, scale up to 3, 4, etc. Okay, so now let's go into some vocab to explain what I was saying before. So we have a homozygote. A homozygote has a pair of identical alleles of a gene encoding a character. This can be shown by the top left here. Let me get the pointer. The top left here and the bottom right here. They have the same character right here. So they're homozygotes because it's capital B, and lowercase b. So you can be homozygous dominant, which is you have both capitals, which that would be purple in this case. Or you could be homozygous recessive, which would produce this, a white character. Then we have heterozygotes, which have different alleles. We discussed that. Then there's a genotype, which a genotype is the organism's genetic makeup, the alleles it contains. For example, the genotype of this plant right here would be capital B, lowercase b. This one, capital B, lowercase b. The phenotype describes the organism's expressed trait, which would mean that this organism's phenotype would be purple, 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 and this organism's phenotype would be white. All right, so now onto the question that was in the chat. What makes something dominant? Like, why does the purple mask the white allele? So for an organism to express a dominant trait, it typically only needs one allele, as it can cover for a non-functional recessive allele. What that means is, we'll see later on too, but these DNA molecules, like I said in the previous class, they're encoding the proteins. So think of, instead of thinking them as like genes right now, think of them as the protein molecules that they encode. So we have the purple one. It produces a pigment that's purple, and the white one produces a pigment that's white. So if we have one purple allele and a white allele, or the variations of the trait, then what's going to happen is the flower is still going to look purple. Because the reason is, even if the organism only makes 50% of what's actually normal, for example, if it was homozygous dominant, it can still produce enough purple pigment for the plant to appear purple. That's why the heterozygotes here still appear purple despite having a white allele, because it's still producing the white protein, but it's just not visible to the naked eye. Secondly, we have, this is why heterozygotes express the dominant allele. So, and that's how you can determine if one's dominant or one's not dominant, by doing this cross right here and looking at the heterozygotes and seeing, okay, if it's purple, then purple is going to be dominant. All right, so now we'll discuss the law of independent assortment. So Mendel, he wanted to spice things up, I guess. So he studied crosses where more than one character or gene or trait was actually being analyzed. So this led to the creation of the dihybrid cross. So in this one, we're analyzing 2 genes now at the Punnett Square. We have round and yellow, which round would be dominant, and yellow would be dominant as well. And round and yellow is also dominant traits. So what we do is we take a combination of all the different types of gametes that could be made, and then we do a Punnett Square with that. So think about it this way. Round, in this case, they're going to be heterozygotes, because you know that the top one, the Punnett Square, has a capital R, has 2 capital R's and 2 lowercase r's, and 2 capital Y's and 2 lowercase y's, which means it has a mix of both the recessive and the dominant alleles. And so therefore, there's 4 different combinations that it can make. If it has a capital R, lowercase r, capital Y, and lowercase y. There's 4 different combinations here. And we're doing the same thing for these ones. And it's sort of like a multiplication table where we match them all together. And then we can determine their outcome based on that. All right, someone asked, what if the parents only have dominant traits? That's a great question. If the parents only have dominant traits, let's say in this example, if it wasn't capital B, lowercase b, but it was just all capital B's, then that would mean that there would be true breeding. That would be an example of true breeding where it would only produce purple plants. Also, the same could happen if it was all recessive. Then it would only produce recessive traits. Does that answer your question? Or yeah, okay, cool. So that's what would happen if they're dominant. All right, so what are the chances of traits from earlier generations of the parent reappearing in the offspring? Okay, so this is how it works. The parents, let's say the mom and dad, they have their alleles from their parents and their parents, because the dad, for example, would give, or the male, I would say, would produce one, or it would give one of its alleles to the offspring for a certain gene, and the female would do the same thing as well. So that's why we make these different combinations right here. These all represent 4 different possibilities. So this the female right here could either give its capital B or its lowercase b, and this one could produce its capital B or lowercase b. So over time, the traits of an earlier generation passing on to the offspring would be 1/2. So the trait, this capital B trait, passing on to the offspring from the mother would be 1/2. Same with this one also would be 1/2. So yeah, obviously these like traits don't just come out of nowhere. I mean, sometimes they can come from mutation, but they usually just come from previous traits in the past, where the mother produces gives one of its alleles, the father gives one of its alleles. They combine to pick an offspring, and the cycle starts all over again. Did that answer your question? Or do you need any more clarification? All right, take that as a yes. Someone said, how do you access the slides? They'll be posted on the Kid Teach Kid website under supplemental materials, I believe. All right. So yeah, so back. Oh, can the recessive trait turn into the dominant trait through evolution? I'm not entirely sure. Okay, I guess through evolution, and from what I understand, it wouldn't necessarily turn into a dominant trait, but there's a lot of instances where a recessive trait is actually more common in an environment. So there's something that people can sort of like misunderstand, is that the dominant trait isn't always like the best trait. Or typically dominant traits usually like the functional trait, and the recessive trait's usually the one that's not functional. And that's why you see like the crumpled seeds right here in this one, where it has recessive stuff. But over time, there's certain examples where the dominant allele is actually harmful, or it's like abnormal. For example, polydactylity, where you have multiple fingers or more than 5 or toes, then they would that's actually dominant allele. But you don't see most people with more than 5 fingers or more than 5 toes. The reason why is that over time, like you said, through evolution, it was more evolutionarily favorable for the recessive alleles to just propagate. And so that's not a question of whether it's dominant or recessive, but it's more of a question of rather like, is it more suitable for the environment? For example, if for a certain trait, blue eyes, make blue eyes dominant in that family. Okay, so in that family, it doesn't make it dominant. Like something's only dominant or recessive, but what happens is in certain traits or crosses, when it combines together, then it would like produce the outcome. So I think a good example of this is back to this slide right here, where I talked about how if they're all dominant right here, let's say these are all lowercase b's, these are all white flowers, then all the plants would be white. That doesn't mean the trait's dominant, but that means that the trait's still recessive, but this means that there's nothing there to mask it. So in blue eyes, we talked about how like that's usually a recessive trait compared to brown eyes. So if let's say instead of purple and white, it was blue for the recessive trait. So even if they're like two parents, they both had blue eyes and they produce offspring and they all had blue eyes, that just means that they were in this corner right here. They both had the recessive allele. But dominant means that if it was a heterozygote, it would express that dominant allele. For example, brown eyes. If it was brown eyes and blue eyes allele, then it would express brown eyes, making it more dominant. Does that answer your question? Or both questions, I guess, about the evolution? But yeah, so evolution and dominance are completely different things. Just over time, evolution can pass certain traits, but the dominance is only really in specific instances. So is it possible for 2 parents to have 3 kids with different color hair? Yes, it's actually possible. So yeah, it's great to ask questions. So let's look at this thing right here. So let's have 2 parents, they're heterozygotes. They have 3 kids, or they have 4 people, and they have 4 kids right here, and there's 3 different hairstyles. So yes, it's possible because of the combination of the alleles that would come together. So let's say they both produce the dominant allele, then that would happen. They would also have like the dominant hair color. then these ones would have the same dominant hair color as well. And then this person right here would have the non-dominant hair color. All right, how do you represent, for example, someone with black hair and another with blonde hair having a child with red hair? So in that case, all right, well, that would be a specific instance, I guess. I can type that in chat actually. Yeah, I'm just gonna type that in chat because it's easier to write down. It depends on how it actually works. I don't know if this is biologically accurate, but okay. I think in this case, there would have to be multiple genes, which would get into something a bit more complicated. But for the sake of simplicity, let's just say that black hair would be dominant, blonde hair is recessive, and red hair sort of an intermediate. Then that's actually, yeah, I'll actually address that later. That's going to be in a future slide. Yeah, that's a good question to ask. So yeah, that was so back on independent assortment. So that just shows that like the different genes here in a dihybrid cross, they can sort of assort independently of the other genes that it's in this gamete. However, the law of independent assortment by Mendel was good. It helped geneticists learn a lot of important things, but when it came to a point where it actually got disproved in specific instances on linkages or on linkage when certain genes would travel as a unit along a chromosome. Could only one parent give both alleles and the other none? Okay, that's also another that's also another topic. I guess we'll talk about it on here because this is where all of your questions are sort of coming down into what we're talking about in this slide. So yeah, I'll get to that question though. So we have incomplete dominance here. This is sort of like the black hair, blonde hair one, but a better example would be whenever heterozygotes have a blending character. So let's say we have a red flower, that's true reading, so capital R, and a white flower, which is also true reading, lowercase r. All the people would have, all the flowers would be pink, because they would get one capital R from their red allele from their one of the parents, and the other one from the like other parent, they would get the white allele. And in this case, there would be a blending characteristic. It happened in some instances where the blend makes a pink one. That's incomplete dominance. So that sort of shows how stuff is blended together, which might answer your question, if we're the one who asked with the blonde hair and red hair, depending on the specific instance of it. But there's another example where that could happen. That's epistasis at the bottom. So then there's codominance, which shows that both traits are expressed in heterozygotes. So let's say we have a white cow and a brown one. They mate together and they produce a spotted cow. So this is how it is because both traits are expressed. It's not like an intermediate traits, like the pink traits being expressed. You have one, you have the brown one and the white one both showing simultaneously. Then you have pleiotropy, which is when a single gene product has multiple different effects. And so that can be shown in cystic fibrosis, which is a disease where it affects a certain protein. And since that protein has so many different effects, there's a lot of different symptoms that can result. And multiple alleles, which is your blood type. There's different alleles in your blood type. You have the A allele, the B one, and the regular one with the O. And so that's another thing that differs from Mendelian genetics. And then this is epistasis. So epistasis is beyond the scope of this discussion. Yeah, it's beyond the scope of this discussion, which does technically answer the black hair and blonde hair one. But think of it, it's suffice to say that let's say we have two genes and the product of one gene alters the expression of alleles at another gene. For example, let's say we had two genes. One gene was to code something that makes either a black one or like a black hair or blonde hair. Okay. And then we have another gene that dictates if the hair will have color at all. So depending on, so the hair can be black or brown, but let's say the hair, the gene that dictates if hair will even have color doesn't even occur or becomes non-functional, then no matter what the alleles on the other gene for the black and brown, it'll be white no matter what. So it shows how different genes have a modifier effect. So in this case, maybe which is black or brown or black or blonde, and another gene dictates sort of like the gradient of how intense it is, which may produce something different like red hair. Then another person asked, can only one parent give both alleles, another none? Actually, wait, no, that's at a different slide, my bad. That would be near the end of the lecture. That's when you get into like meiosis. But yeah, that does happen. All right, so this is an example of the blood system in humans. Only press answered. on this. But this is an example of the blood system. So we have different alleles here. We have the IA, which is for A blood type, IB, and I. I'm not getting into too much specifics. When I post the slides and hopefully the problem sets, I didn't post the last ones. I'm going to put those, sorry about that, for the last class. But then you can probably explore a bit more how the blood types actually work together, because we have a lot to cover in a shorter amount of time. But there are multiple alleles for blood types and they can combine together. So yeah, you have the A blood type, AB blood type, and the O blood type. It's important to know the blood typing systems because whenever people are like donating blood, if certain blood types are incompatible with each other, then it can produce some pretty bad outcomes. All right, so now that that's actually good timing. So for the first half of the class, we discussed sort of like the conceptual basis of genetics, how genes are passed on, etc. Now we'll transition into the molecular aspect of genetics. So on the last class, we talked about how like DNA, proteins, and macromolecules and how they all work together. And they also work together hand in hand with how we transmit our genes to offspring. That's like the interplay between biology, biochemistry, and genetics. So this is called molecular genetics. It deals more specifically with DNA molecules and nucleic acids and how these molecules produce the gene products and proteins that travel within our cells. So here we have an example of the double helix structure that we talked about last time. And we have the different bases here. So yeah. So to start off our talk on molecular genetics, we'll talk about chromosomes. So I know I mentioned the word chromosome a lot, but let's talk about what it actually is. Think of chromosomes as the functional unit of the inheritance. So, or that's actually a gene, but it holds a bunch of genes together and it transmits that to offspring. So genes have certain positions along these chromosomes or loci, or that's the position of a gene on a chromosome called a locus. And the chromosomes are also a compaction of these really long DNA molecules associated with proteins that are needed to compact them. So what do I mean by this? I remember if you were to stretch out the DNA molecules, they would stretch out over like many miles, like some of the chromosomes, they'd stretch out over many kilometers. And the cell nucleus, which has to fit in, is only a couple micrometers. So it has to fit in a ton of stuff into a very small amount of space, which it uses these proteins to compact all the stuff together. These proteins are called histone proteins. I'll type it in chat actually. Yeah, they're called histone proteins. And histone proteins are needed to compact all these DNA molecules together into chromosomes inside of the nucleus. So like I said, chromosomes are what's transported to offspring. And chromosomes are the units that undergo segregation and independent assortment through the recombination, which we'll also talk about in the meiosis section. So we have 23 chromosomes, as you might know, or technically 46, because we have 22 autosomes and 2 sex chromosomes. However, we get one set of them from our mother and the other set from our father. So we really get 20, we really have 46, because we get 22 autosomes from our mother, 22 from our father, 1 sex chromosome from our mother, and 1 sex chromosome from our father. Females, and regarding the sex chromosomes, they have 2 X chromosomes, and in males, we have 1 X chromosome and a Y chromosome. Yeah, so yeah, here's an example of a chromosome. This is not the actual right picture. I'll find a picture of it, but how we show the chromosomes is on a karyotype, and like we organize them. Let me find a picture. Yeah, this is technically right, but so this is an example of a karyotype right here. You can see that, you can see how the chromosomes are just arranged here. And so what happens is they find these chromosomes in the cell, scientists, and then they order them based on their length. So you can see the chromosome number 1 is really, really big, but then chromosome 22 is smaller. So it's just ordered based on length. And you have the X and Y chromosomes. But yeah, that's chromosomes right here. So yeah, now let's talk about cell division. So there's 2 types of cell division. We have mitosis and we also have meiosis. So mitosis describes cell division in our normal body cells. For example, our skin cells, our bone cells. That's mitosis. And what happens is in mitosis, you have a normal cell that's diploid or diploid. What that means is it has 2 sets of chromosomes, one set from its mother, one set from its father. And so they replicate, and it produces two new cells. Each one of those cells is genetically identical to each other, and it genetically identical to each other, where one of them has one set from its mother, one set from its father, and same with the other one. But meiosis is a bit different. So it starts with one cell, same as mitosis, starts with one cell that has two sets of chromosomes, that's diploid. But then it produces four genetically different cells with one set of chromosomes each, or haploid. We call cells with one set of chromosomes haploid, and cells with two different chromosomes or two sets of chromosomes diploid. So when this happens, like why this happens? So we'll talk about the specifics of the stages of mitosis first to get a better understanding of how this occurs. So mitosis, before we actually have cell division, we have interphase. Interphase is whenever the cell grows and duplicates its DNA. So the reason why the cell is able to produce two new cells with each the same amount of DNA molecules, right, is because the DNA doubles. So originally, let's say we have the 46 chromosomes and the two sets of 23. Before mitosis, it's going to double to 92, and then it's going to divide into two cells, each with the same 46. So the 5 stages here, I'm not going to get too much specifics about them. We have prophase, metaphase, anaphase, telophase, and cytokinesis. Suffice it to say that the overall gist of it is you have these chromosomes here, and originally in interphase, they're pretty not condensed. They're just they're sort of like onion just floating around in soup. That's the best analogy I could give. Sorry. And then furthermore, later on, like metaphase, they start to order on the middle of the cell. And in anaphase, they separate from each other. Or so the chromosomes like the X, right? They have a sister. So these two things together are each chromosomes, right? They're the sister chromatids. So each chromatid gets pulled apart from it. And so then you end up with the two cells that have the same amount of genetic material because they check half of something that was doubled, meaning it's the same. Because that's the main gist of mitosis. So now let's look about meiosis. So then I'll get to your answer about our question about the one parent producing nothing when one parent producing both alleles. So the difference between mitosis and meiosis is that meiosis divides twice. That's why 4 cells are made. So instead of 2 cells. So here we have 2 cells. If each one of these cells divided again, they would produce 2 cells, or 4 cells. But then you might be asking here, well, wait a minute. Why doesn't the DNA still remain the same in meiosis? Why are they haploid now? The reason for that is between this first and second division, the DNA doesn't replicate again. So then it just splits in half. You can see this right here. Oh, by the way, the red things right here are the father's, or in this case, we'll just make it like the father's chromosomes. The blue ones, we can represent them as the mother's chromosomes. So what happens here originally in mitosis is that you have, or in meiosis, we have firstly the two of the one parent's chromosomes separating with each other across a different part of the cell, and then those things do the same thing again in meiosis 2, which is the second phase, which then it produces 4 haploid cells again. So why are the cells genetically different? Even if let's say the DNA replicated, the reason why is we have the stuff from our mother and our father that are from our mother and our father, and under meiosis, sometimes they can undergo recombination. Besides the fact that the mother and father could have had different alleles, recombination occurs where, let's say, these chromosomes exchange different pieces. So yeah, they exchange different pieces of themselves, which is why you have some chromosomes that are both blue and red. All right, then, so yeah, now the question, can only one parent give both alleles and the other none? So yes, when this occurs, we call it non-disjunction. What I mean by non-disjunction is that, well, it's in the name. Disjunction means separating, and non means it's not happening. So let's give an example here. We have, let's say we have the mother cell, and we have, actually, let me try to get a drawing. Yeah, let me get a drawing out here. Let me just get, this snipping tool, something to draw on, I guess. All right, so let's say we have the mother cell. It's gonna be some pretty bad drawings, but it's fine. We have this cell right here, which is from the mother, and then get a blue. This cell right here is going to be from the father. And so, yeah, we have the 2 different gametes here. Let's say our mother, in meiosis, the chromosomes don't actually separate properly. So we have all of our chromosomes like chilling here. And then we have one cell. So we have the cell separating in two right here in this phase. But let's say like this chromosome right here accidentally pulls both of them up here and leaves here, this one without a gamete. Or just like all these three things right here just get pulled to this core. This pull and this one gets nothing. And so it would divide again. And when it would create the different progeny, it would create one cell that has nothing. Then the three other cells that have a certain amount of chromosomes, like XX. I'll draw some X's here to show they have chromosomes. And so when we have this gamete right here, let's say out of the gametes, biology is interesting. So yeah, we have one of these cells right here that doesn't have a chromosome. And then we have a father's one, for example, that just has an allele. It's a normal one where it just has one of its X's. Then it would create a cell that actually has only one, not two. And that's not, let me make it green, or actually purple. Yeah. And then we'll create a cell where it has only the father's blue X. But let's say, and something else happened where the father, so usually what happens is in meiosis, we have like let's say we have 4 X's right here for 4 chromosomes. It would produce 2 cells like this, where this would be 2 X's, 2 X's. Then this would produce in one cell with an X. But let's say in this one where we have 2 X's here, it accidentally produces one that has 2 X's and another one that has 0 again, right? Similar to this situation with the mother. But this time the 0 chromosome comes here and the 2 chromosome or the 2 genes come here. That produces a cell that has 2 from the father and none from the mother. So that's how that would work out. So it's really complicated how all of it comes together, but that's non-disjunction. Does that answer your question? I know it took me a long time to answer that, but yeah. Yeah. Oh yeah, by the way, if this is confusing at all, if I'm like explaining this badly, the slides will be posted if you just want to go over them again. And obviously, like you can do a bit more. If you're really interested in this, I recommend doing a bit more research on it because really only has so much time to cover like a lot of really like in-depth material. So yeah, that's what happens there. And then a bit more on recombination. So we talked about how the chromosomes exchange the material once again. And so this is how the genes would independently assort. So let me just like, okay, I have to erase this. All right, I'm gonna just erase this or just take a new snippet. All right, so here, let's do this again with the chromosomes. We have, make it blue and red this time, or aqua. So we have the mothers. Let's get the fathers here. Yeah, it's red. And so in this case, let's say these 2 chromosomes. So let's say like the right one on the blue and the red one here, they just like came together. So this happens in prophase of meiosis. So we have this one right here. Let's just like zoom this in a bit more in depth. So we have these 2 ones right here. They can exchange chromosome pieces during prophase 1 of meiosis. And so let's say like these 2 things like exchanged. This part would become blue and this part would become red. They exchange the pieces. And so then this is how it gets passed on like that. So this actually explains independent assortment that Gregor Mendel made up, because it shows that even though the genes are on the same chromosome, they can go transport to other chromosomes. They can just like go to a different one, get transported off there to the offspring. But sometimes what happens is, the reason why I talk about linkages and how like sometimes the actual genes are transported as a unit together, like a pair that you cannot separate, because of linkage. Let's say we have like okay, so we have let's say this is 1 chromosome right here, okay, and we have these 2 different genes. This like each marking represents a gene. So the chance that a recombination occurs between these 2 genes right here, it can be very low because they're so close to each other that it would have to exchange such a very minute section out of a whole chromosome which it could be exchanging with. But if we look at these 2 genes right here, there is a much higher chance that recombination is going to occur. So yeah, therefore, yeah, there'll be much higher chance that recombination occurs because like it would much more ground to be covered for the different chromosomes to cross over. Something interesting with this that it's not necessarily important to know, but it can get covered in a lot of really important topics such as USABO, is that whenever you do a bunch of crosses together and see how two different genes, like the recombination rate, there's a way you can measure that. But with that, since a higher recombination rate means that the genes are further apart on the chromosome, a lot of scientists can actually map the chromosome to see which gene is, or like where it is. And so that's how different genes are located in specific loci in the chromosome. They measure their recombination rates, amongst other ways as well. That's how they originally did it. Now there are more high-tech ways like actual gene sequencing. So yeah. All right. I know that was a lot to cover. So if anyone has any questions about like the mitosis, meiosis stuff, you can just put it in chat. Because we only have a couple more sections left. Yeah, we have DNA replication, then we'll do a quick overview of protein synthesis. All right. All right, I guess we can maybe, okay, can you state the difference between mitosis and meiosis? All right, cool. So mitosis, we talked about how it was in our body cells, and meiosis occurs in your sex cells. So mitosis is what makes us like us. It makes our skin. And it doesn't, and stuff in meiosis doesn't actually transport to like your future offspring, but the meiosis produces the gametes that fuse together with another organism, or hopefully like a human, that would create the offspring in this situation. So yeah, also mitosis produces 2 diploid cells. Meiosis makes 4 haploid cells. Okay. All right. Well, it should. All right. Let's not get into that conversation here. Okay. So on to DNA replication. So DNA replicates via the semi-conservative model of replication. So what does this mean? I may have forgot to actually write text on this slide, but we can just talk about this diagram. And it's actually really cool history of how scientists figured out how DNA actually replicates. So it's actually a good conceptual start to it. There's 3 models here. Scientists didn't know which one is right. You had the conservative model, the semi-conservative model, and the dispersive model. So the conservative model was you split the strands together, you make new strands of DNA on them, the new strands combine together, and the old strands still stay with each other. So that's how conservative is. It conserves the original DNA strand. Semi-conservative means you sort of conserve it, but it's not like it's not like you're losing this, like one of the strands entirely, but it's just like mingling with another strand. So you have here the 2 strands separate, then you add DNA to the first one, making a new strand, which is one old and a new one, and then you do the same thing again. So you break them apart and you make a new strand with it. Then the dispersive model is just chaos, where I don't even understand how scientists come up with this, but you have different parts that bind to it and replicate, and then it just creates a whole different like meshwork of it where the DNA disperses to different DNA strands. So scientists are like, how do we know which one's which? Obviously now when you think about it, the semi-conservative model just makes the most amount of sense. But scientists found a really clever way to figure this out. What they did was they took advantage of something called radio labeling, which they use a radioactive molecule, and then they tag it to the DNA strands. And so they can visualize it based on certain techniques. So what happened was they grew the original DNA strands with a certain radio label that had a certain radioactive molecule placed to it. And then what happened was they grew it and they made sure that it was just those things over a bunch of generations. And then what happened was they put it in an environment where it had a different radioactive label in it. And so the new strands that were being made would have that radioactive label. And over time, they measured the amount of radioactivity of the certain molecule that they originally put in over time. And so their experiments, when they did it, at first they're like, okay, oh yeah, I'm trying to explain this. All right. But yeah, so first, whenever their data came out, it matched the semi-conservative model based on the amount of radioactive ones from their original environment and based on the new environment. They realized it was a semi-conservative model of replication rather than the conservative or the dispersive model. So yeah, because they would each find individual DNA strands with this amount. Yeah. So now let's actually talk about how DNA replication occurs. So, like we said earlier, the DNA strands, they separate, and RNA, and then DNA polymerase, which is an enzyme. We talked about enzymes in the previous class. They're the proteins that catalyze faster reactions. They add the appropriate complementary bases to the strand. What I mean by that is, let's say the strand of the base is like adenine, it's going to add thymine to that, because adenine pairs with thymine and guanine pairs with cytosine. So DNA polymerase reads the DNA, like, okay, that's an adenine, I'm going to add a thymine there. And then the replication starts. Another thing to know that the replication starts at a point of the DNA called the origin of replication. And this is a good thing to know as well. There's two different strands, because it's double helix. The top one here is going to be the leading strand. The bottom one here is going to be the lagging strand. And so in the leading strand, it's like the DNA polymerase just goes, all right, I know. Okay, so it's like, I always make this analogy. I don't know if any of you all play Minecraft, but there's the term in Minecraft called like God bridging, where someone's trying to like make a bridge in Minecraft and they're going insanely fast. And this is how it is. Like the DNA polymerase is just going insanely fast right now, adding a bunch of DNA molecules. And then in the lagging strand, which is the bottom one, it's sort of like it adds short bursts of different molecules. And then it's like, so yeah, adds short bursts of different molecules, then it fills in the gaps over time. The idea of like you can see the leading strands going like much more quickly here. And yeah, yeah. I mean, okay. I mean, people don't play Minecraft, so yeah. Like I only recently knew what God bridging was, but that's besides the point of the lecture. So yeah, I figured that a lot of y'all probably play Minecraft. All right, so I guess we'll end off the lecture with protein synthesis. So protein synthesis, this describes the central dogma of genetics. So central dogma of genetics is you have DNA that goes into RNA, and that becomes protein. So there's two different like sections to it. You have transcription and translation. Let's relate this to something that y'all might be familiar with once again for the what's it called? Yeah, the USCO class y'all just went to, the coding. So think about it this way. You have DNA, which is the code that you put in, and then it gets transcribed. That would be sort of like the compiler. The messenger RNA, or in the RNA polymerase, is sort of the compiler that turns the DNA into mRNA or messenger RNA. And it turns it into a language that machine code can understand. And the machine code in this case would be the protein, or not protein, the ribosomes. The ribosomes are like the machine factories that translate whatever code the mRNA is into a sequence of amino acids, which is in the past discussion, if you want to see the slides about amino acids there. And based on the mRNA sequence, it creates a bunch of different amino acids. This is called the genetic code because it's basically a code. You have these ribosomes and RNA polymerase, which is reading these sequences of DNA, and it's creating RNA strand and amino acids. Okay, so this is a picture of it, double-stranded DNA, and you have the messenger RNA being made. The RNA polymerase works very similarly to DNA polymerase, except that it only really needs to replicate one of the strands. And it has uracil instead of thymine. And so over here you have the ribosomes, which it just reads these reads the RNA code and then adds the amino acids to the protein, which then makes the protein. All right, that's the end of the slides. So now I'll be open for some more questions. If anyone also wants to go back to a previous slide, just say that as well. Or if they have any like concerns or comments about this lecture, feel free to say them. Someone asked, where can I find these slides? They'll be found on the Kid Teach Kid website. They'll be posted sometime after the class, but you can even see the last slides. So in the last class, if you go into class resources, you can find the recording, and under supplemental materials, there's a PDF that has the slides you can download. Good question. What is happening in the ribosome? Okay, so this is also beyond the scope of the discussion. You can research it a bit more in depth, but let's just give like an overview. So what happens is we have the mRNA here that goes to the ribosome. So the ribosome sort of like is a machine. It just like holds it together and it reads the RNA code, but it reads it in terms of 3. So every 3 things right here, like A, U, G, that represents something called a codon. And it sort of groups them together. It's like a statement. And that codon's like, okay, that directs this synthesis of a certain amino acid. And the reason it does is because you have these other RNA molecules called tRNA or transfer RNA that contain, let's say, an amino acid like serine or threonine, glycine, leucine, or methionine. So it carries these amino acids, and it also has a sequence here that binds to a specific codon. So let's say we have CUC, that would bind to GAG on the tRNA, and that carries leucine, which means that CUC would specify leucine. So the tRNA binds to the, that specific site, and then it releases its amino acid that it holds onto to the polypeptide chain that's growing along the ribosome. There's a lot of complex stuff, but I definitely recommend that you research about this more in depth and pretty much about all the topics we discussed here today, because we really only had an hour to discuss a lot of stuff here. What does the polypeptide chain do? All right, cool. So in the last class, we discussed about how the proteins are made from these peptide bonds. So we have these amino acids, they bind together to produce peptides or proteins. So the polypeptide chain makes a protein basically. So it folds together in the way that we described in the previous class into a fully functional protein. And these proteins direct all the bodily functions or all the cellular functions that occur. For example, the pigment in the flowers that makes it purple versus white is a protein. The pigment is a protein. Or something that a storage protein. An enzyme, right? All enzymes are proteins. So these polypeptide chains can become an enzyme based on the sequence of amino acids there. And how, like, the sequence of amino acids determines how it folds, its properties, etc., how it reacts in certain temperatures, environments, and that can specify its function, for example, if it wants to become an enzyme. So that polypeptide chains, that's how like the genes turn into actual action for the cell. So yeah, that's why we say the nucleus has the instructions to make the proteins. Yeah, that's what the DNA tells cells. Yeah, so yeah, it's really cool. A lot of people might just say DNA nucleus tells the cell to do stuff, but actually what really got me hooked onto biology too, when I realized how the DNA actually does all these different functions. It's really cool. So yeah, but there's a lot more in depth about how this stuff actually works that we couldn't discuss in today's lesson. So if you want to research or you're like even mildly interested in how it works, I would definitely look into it. Any other questions? We have like 3-ish minutes left. Also, if you have any questions, put them in the Q&A. Oh, someone said earlier, well, I don't understand the slide. Wait, which slide were you talking about? Sorry, I didn't see that message. Or did I already answer it? Oh, you explained it? All right, cool. My bad. I was looking at the chat. Any other questions or? It's good. All right. So if no one has any other questions, then what we can do is we can just end the class a bit early. I'll stop screen sharing and I'll transfer co-host to Tanya, and yeah.